Somewhere inside your body right now, there's a protein behaving like a quantum computer. Not in a lab freezer, not near absolute zero. Inside warm, wet, chaotic living tissue. For a full century, physicists lived by one unbreakable law. If you wanted to see quantum effects, you had to freeze your system down to temperatures colder than deep space. That's why Google's quantum computer sits inside a giant gold chandelier of wires, and IBM systems need entire rooms just for refrigeration. These machines are unbelievably powerful, but also unbelievably fragile. Then, in August 2025, a team led by Awschalom and Peter Maurer at the University of Chicago's Pritzker School of Molecular Engineering did something nobody thought was possible. They took a glowing protein, the same kind that makes jellyfish light up green, and turned it into a working quantum bit, a qubit. And it worked at room temperature inside living mammalian cells and bacteria. The paper was published in the journal Nature. Physics World named it one of the top 10 scientific breakthroughs of the entire year. The protein they chose is called Enhanced Yellow Fluorescent Protein, or EYFP. It belongs to the same family of glowing proteins originally discovered in jellyfish. The team found a way to excite this protein with a 488 nanometer laser and then read out its spin state optically. In simpler terms, they found a way to ask the protein a quantum question and get a quantum answer back just by shining light on it. They achieved 20% spin contrast at cryogenic temperatures and a 16 microsecond coherence time. And critically, the quantum behavior carried over into living cells at room temperature, with contrast up to 8% in E. coli bacteria. Protein qubits are different from anything that came before because they're made of, well, protein. They can be genetically encoded directly into the DNA of a living organism. That means the cell itself builds the qubit. It manufactures its own quantum sensor, positions it exactly where researchers want it, down to the atomic scale, and keeps producing more as it grows and divides. Biology doesn't make one or two copies of something. It makes trillions upon trillions of identical copies, something engineers building quantum devices in a lab could never realistically achieve. So what do you actually do with a quantum sensor that lives inside a cell? One of the biggest applications researchers are already discussing is quantum-enabled nanoscale MRI. Imagine an MRI, but instead of scanning your entire body, it can map the inside of a single cell, atom by atom. Individual molecules, individual proteins, mapped with a level of resolution current medical imaging simply cannot reach. Misfolded proteins are directly linked to Alzheimer's, Parkinson's, and many cancers. If scientists can eventually watch these folding processes happen in real time inside living cells, doctors could detect the earliest molecular warning signs years before symptoms ever appear. To be clear, this is not a full quantum computer sitting inside a living cell running complex algorithms. What researchers created is a single functioning qubit, a quantum sensor. Photo bleaching still needs solving. Readout fidelity still needs improving. But every quantum computer ever built started with the challenge of creating one working, reliable qubit first. This breakthrough proves, for the first time, that a living biological system can produce and sustain one entirely on its own, using its own natural machinery. And once a wall like that cracks, even slightly, it tends not to stay standing for very long.